Good afternoon and welcome. I hope you enjoy your sandwiches. Few announcements before I introduce Nima. We have very exciting talks coming. Next week, we have representatives from DHO, the big hedge fund. They're going to be, though, talking about simulation biological systems. You can hear me. And then follow that, we have Ashish, who is going to be talking about interdisciplinary research, astronomy, and machine learning, following by Chris Wiggins from New York Times, talking about data science they've done at New York Times, and two other talks. Let me not take all the time. You can find the talks at our website. If you Google IACS seminars, you'll find it. So it is today my pleasure to introduce Nima Dehghani. Did I say it right? Yes. Nima is a fellow at the VIS Institute, working on neuroscience, working on the brain. He did his medical degree in Iran, came to United States, did some research on the system, this system. I'll say a little bit more. And then he decided that the medical field is exciting, but not as exciting as doing research. So he went and did a PhD in France and back to United States. And his specialty is computational neuroscience. And he's talking about
first line is good, I guess.
Computational networks in neuroscience. And because he was coming, I got a new toy who follows up my sleep pattern, so I want to learn how to interpret my new data. So, Nima?
Okay. Thank you. Thank you. Thanks for the introduction, and thanks for inviting me here. And thanks for the audience to be here. So, you can't hear me? It's on. Let me see. Can you hear me? Is it better? Okay. All right. Okay. So today I'm going to talk about a variety of topics, actually, but they all fall under an umbrella, which is basically we try to understand if the frequency scaling and the avalanche dynamics of the system. You can't hear me again? Okay. All right. So, sorry for the interruption here. So, I'm going to talk about computational network dynamics in the cortex from different points of view. And that actually includes a variety of topics of looking at ensemble activity of neurons. So, for doing so, we actually did these kind of experiments when I was in France in the Laboratory of Computational Neuroscience, and also part of it while I was here in Harvard. So as you can see in the top, there are a few different segments in the talk, and including spectral dynamics, avalanche dynamics, spatiotemporal dynamics, and balance dynamics. So today I will get to talk about like part of them, but they all basically are connected to each other within a bigger framework, and that is, for example, the idea of brain as a complex system is very much an interesting idea because, for example, we see a variety of scientists trying to look at structural connectivity or functional connectivity, where they look at the wiring of the brain with the diffusion tensor imaging, and they try to relate these different concepts together. So that's in basically a human brain. And there are some other scientists who have, worked on C. elegans, which is a worm, a nematode, and it has only a few hundred neurons, and we know the exact wiring of these neurons, mainly due to the work of Sidney Brenner, who won a Nobel Prize in molecular biology. But he and his colleagues did this kind of work. But still, we don't know what's going on. Like, how does this simple system operate? And similar kind of things, trying to find the exact connectome, the exact wiring, all of the connections in a given system is also being done in other animals, like zebrafish. That actually partly is being done here at Harvard. Perhaps you have heard of it under the umbrella of connectomics. So there are some who argue that brain is like a small world network. The idea is that you have a structural dynamic and the functional network dynamics, and the connection between the two of them happens either if the system has a sparse coupling, high order or low complexity, or uniform coupling, high order and low complexity again. So these are two possibilities for the system. There's a third possibility, and that is that you have local global coupling in the system within your network, but also you have a mix of order and disorder, which rises to higher complexity. So some argue that this system can actually explain the complexity as is observed in the brain. And some follow that argument and propose that, therefore, the self-organized criticality plays an important role in the nervous system. So let me just talk a little bit about self-organized criticality if someone is not familiar with it. And that's the idea like this. So imagine you have a bunch of magnets. And this is the orientation of the spin of the magnet. So if they're all like ordered in low temperature, they point the same way. If it's high temperature, they are randomly oriented. And there is some point, there is some critical temperature within which you have some sort of like complex set of orientations. Like it's somehow ordered, but also somehow disordered. So if you look at it, for example, if this is the temperature and this is the correlation length of the different elements in the system, here you have order at low temperature, and here you have disorder at high temperature, and this is the phase transition point, basically, around the critical point. So at this moment, the system could fall either to complete order or complete disorder, and some argue that being here is beneficial for the computational principles that could the system benefit. For example, some argue that it actually helps in increasing the information capacity and such and so. There are some caveats about this kind of thinking. For example, if you look at certain oscillations, so brain always like has certain types of oscillations. For example, when you sleep, in the stage 2 or 3 of sleep, which is somewhere between the light sleep and the very deep sleep, there's certain grapho element, there's this oscillatory thing that shows up across the cortex, and it's about like, half a second to 2 seconds long, and its range of frequency is between 7 to 15 hertz. In actually smaller mammals, it's in lower frequency range. And when you look at it, a given spindle, if you look at the activity of it, so this is the magnetoencephalography of the whole human cortex, and this is some sort of like inflation of the cortex, imagine, for finding out the whole system. And here you can see every 20 milliseconds, you see a snapshot. This is the left hemisphere, this is the right hemisphere. And the inflation, if it's not familiar to you, the tip of the frontal lobe is here, which is right here. And the back of the occipital lobe is here. This is parietal lobe, and this is the temporal lobe. So you're looking at my left hemisphere, for example, and this is the opposite, so facing the other way. And you can see that a given spindle moves around within few hundred milliseconds, drastically moves around. It never actually tries to propagate to the same places. Well, the idea previous to these kind of experiments was that when you have spindle oscillation, then the whole brain becomes active at the same time, and you have massive synchrony, which is more or less similar to what I was telling you before. And then, how do we reconcile the concepts of local global coupling as well as the complexity that you see in the oscillations? So there's an argument. I'm not sure if this is very visible to you or not, but I can just tell you that there's a different sorts of behavior that the system can show. And they range from synchronous regular, which is here, to asynchronous irregular, synchronous irregular, and synchronous irregular. And all these behaviors could be basically shown with some sort of interaction of stochastic dynamics of excitation and inhibition. So that's one of the fundamental properties of the brain. The brain actually is composed of two opposing systems. You have excitatory neurons and you have inhibitory neurons, and the interaction between the two of them could give rise to such rich behavior. Okay, so next, I'm going to jump. So because of shortage of time, I wouldn't talk about spectral dynamics. And I would jump ahead. Okay, I'll jump ahead to invasive recordings in humans. So the main reason is that we do benefit from invasive recordings. Some people do the experiments in vitro settings, which is problematic because you are dealing with a system which is in low conductance state. In vivo brain, you have high conductance state. So the whole system is under current bombardment by the barrage of synaptic activity of the other neurons in the network. So it's very important to do these kind of experiments. So we were lucky to get a bunch of data from collaborators in humans who are doing these experiments in humans. So the experiments are done in epileptic patients who have intractable seizures and they go under long-term monitoring. Or they were done in monkeys and cats. So what I'm going to discuss today is about these kind of data. And they usually are based on 100 electrodes within a 10 by 10 microelectrode array. And it gives us the ability to assess the activity of a bunch of neurons at the same time. This is, for example, an image of such an electrode, which you can compare to the size of a US cent. As you can see, it's pretty tiny. And so this is the big usual electrodes that they put in, and you can see this kind of big, huge electrocorticogram that they put in human patients. And underneath this, for example, here, in between 4 of those contacts, they put one of these electrodes, a multi-electrode array. And it basically is like a thumbtack. So it goes down to different layers of the cortex, and of course there's destruction of the cortex, but it has been shown that after a few days of waiting, then the inflammation goes down, and you can actually see the behavior of the system properly. And here you can see, for example, that how it could be registered to MRI, and you can find the exact location of the electrode. So to the right, you see an example of recording done with such electrodes, which is shown in, so these are the local field potential, and that means that, you're constantly recording, and the low frequency range of it is called the low frequency potential, which is considered to be a signature of synaptic input. And then you have the spiking activity. That's the individual neurons firing, that we turn it into a 1 and 0 binary kind of matrix. And that's what you see here. And that's what we're going to talk about today. So that's the higher frequency part of the system. Let me jump into here. Okay. So what we do is that we take the recordings. So each spike has a special spike waveform shape. And based on the spike waveform shape, you can define if a neuron is excitatory or is a putative excitatory neuron, or inhibitory, is a putative inhibitory neuron. And this is done before in rats and human in the temporal cortex. And we basically use that kind of concept, which is basically a morphological characterization of the system. And we find, for example, the two group of neurons, some fire with a fast rising spike waveform and then a faster decay, and those are called faster spiking. Those are the putative inhibitory neuron. The other group, which is the regular spiking, that's the one that rises to the maximum activity later and also decays later. So this is a spike waveform characteristics of the two groups. This is the clustering of the two systems, and you can do a variety of different sorts of clustering based on these sort of morphological characterization. And you get a pretty robust behavior of these two clusters. So this is only morphological characterization. And when you look at these individual neurons, these individual groups of neurons, so the fastest spiking or inhibitory ones, they usually are lower in number by a ratio of 1 to 4, but they fire faster. So the firing rate of the fastest spiking is more than the regular spiking. And then the coefficient of variance is also rather different. And there are also times that you can look at the autocorrelation of the two sets of neurons. And those with faster spiking behavior, or inhibitory neurons, they have a wider autocorrelation, meaning that they might have some sort of like a memory of the system for longer term relative to the excitatory ones. So, but we also examine how do they functionally relate with each other. So we create some sort of cross-correlation between the spiking of the two systems. And in here, you can see, for example, the two neurons, the one on the left was categorized as an inhibitory neuron based on its morphological characteristics. The one on the right was characterized as an excitatory neuron based on its morphological characteristics. When you do this sort of cross-correlation, within the short latency, like around few milliseconds, you can see when neuron 1 fires, neuron 2 gets quiet right there. So that shows that neuron 1 has some sort of downstream inhibitory effect on neuron 2. And vice versa, when neuron 2 fires, then you can see an extreme sharp activity in the population in the activity of the neuron 1. So it shows that neuron 2 excites this neuron. So these two neurons, one inhibits the other one excites the other. Okay? Here is a unilateral relation of two neurons, for example. And you can see only when neuron 1 fires, neuron 2 starts firing, and there is no inhibitory effect. So because it doesn't pass this sort of like statistical significance threshold. And when you do these kind of things, this pairwise correlation, and you try to map it to the morphological features, you can see all the neurons which had downstream excitatory activity, they fell in a category that had the wider spike waveform characteristics, and the other types of neuron fall within the other category. So this is just the basis of some of the concepts I want to talk about today. Then we look at the activity of these neurons. For example, here in a grid of in a 10 by 10 electrode array, here is an activity of one neuron with respect to all the other. So this is one excitatory neuron. And we categorize its correlation with all the other measured neurons which had excitatory characteristics. And what you can see is that this correlation here is color-coded. And as it fades out, the correlation drops down. And what you see is that as you go further and further away, the correlation goes down. In contrast, when you look at an inhibitory neuron, that's not the case. So you have a variety types of correlation. For example, these two neurons are within the same distance from this neuron. But the correlation between that one and the reference neuron and the second neuron and the reference neuron are vastly different. So this is just one example. But when you take into account all of the activities, for example, for 50 millisecond binning, okay, and this is distance versus the pairwise correlation, then you can see that in the inhibitory category, you kind of like get not much of an interesting relationship. In an excitatory, you get some sort of like a drop down. And you can characterize this by some sort of exponential decay. And you can also extend that to different lengths of binning. So it depends, like, it really depends how many milliseconds you take into account in your bin, right? So we do that, and you get the same sort of behavior. Though you get also some sort of like a spatial modulation that drops down as the binning length increases. So together, what we say is that you can use the morpho-functional characteristics to categorize these two sets of neurons. There's a marked exponential decay in the excitatory category, but not the inhibitory category. And the amplitude of that decline is dependent on the time scale that you're taking into account. And this spatial constant is compatible with typical size of the human columnar organization. So it has been shown actually by David Hubel and Wiesel and a bunch of others that there is some sort of canonical structure within the cortex of the mammalian nervous system. And that is you have a column within which the same sort of computation happens. And then as you move from one column to another, then this type of computation changes. So next, I'm going to talk about the avalanche analysis in the system. Okay. So remember I talked about the self-organized criticality. So that is, if you have a system, within a given period, you have a barrage of activity coming in. And then if you take that into account, for example, if you bin the system with 4 millisecond windows, and you take the size of this window as the number of spikes that appeared within that period, so for example, here you have 1, 2, 3, 4, and then here you have a lot more, such and so, then the distribution of these follows a power law. So meaning that if you plot a logarithmic scale, and this is the length, the size of the frames, and this is the number of avalanches per each bin, then it drops in a straight line. And that actually is a representation of a system with self-organized criticality. This experiment was done in vitro, where you don't have constant bombardment of the system. So there is such evidence in vitro. And since then, a lot of other experiments tried to repeat the same thing. And some argued for it, and some argued against it. For example, here, there was an argument that if you create some sort of function, which is a synaptic current estimated from an exponential decay of the aggregation of your spiking activity, then you can see that the two different states of the brain behave differently. For example, slow wave sleep behaves differently from wakefulness. So such arguments between the two sides had continued. In simulation studies, for example, the one at the top, there was a paper that showed in the simulation of certain sort of a given sort of connectivity, you can see such behavior. In the bottom, you can see that sometimes even a simple stochastic process could lead to the same behavior of, but no power law in the system. And what's important to say is that, so if a system maintains itself close to the phase transition point, then you may have a self-organized criticality. And such system, one of the avalanche, the avalanche of the activity, it follows this rule, so which is very simple, meaning that the system shows some sort of like a scale invariance, meaning that you have the size of different barrage of activity, and the ones with the smaller activity are very much abundant. The ones with the huge number of neurons are not that common. So we look at the system. We look at not only the spiking, but also the local field potential. So here, for example, you can see you can bin the system in two different ways. Those with the red. So imagine that once you bin it with the green ones. So you take into account a longer period of the activity. And then any activity that falls within two sets of them is counted as an avalanche. And then you count the numbers. And you can also, so you get 4, 33, 10. And the difference between these guys is that there's like a gap, a momentary like lapse of firing between these guys. And then on the top you have for another sorts of binning. And you can also do the same thing by discretizing the local field potentials. You get the peaks of the local field potential, and you do the same thing. And what we can see is that when you look at different systems, whether it's a cat or human or a monkey, it's a motor cortex, primary motor cortex, or premotor cortex, the behavior never shows a straight line as they've shown before. So you can see, for example, where you have to take into account is after the green line. So for some statistical reason that I'm not going to go into. But what matters is that the system shows a variety of different behaviors, and it never shows a constant scenario. And when you look at different states, so that was wakefulness. When you look at the slow wave sleep, or you look at REM, which is rapid eye movement, or the dreams period, again, you don't find any evidence for the power law behavior, and therefore there is no signature of self-organized criticality. But when you look at, yep.
This is not the probability of X, is the CDF, right?
Okay, yeah, this is the CDF. So the reason that actually we have CDF is this. When you take, usually people, for finding out if something follows power law or not, they just do a log-log plot of probability and the X. And the issue with that is that you miss certain statistical significance. So if you do a CDF, there's a very nice paper by Mark Newman and Aaron Closet that they go into a lengthy conversation about why it matters to use CDF instead of PDF. The CDF also shows same kind of thing, but then you have to take into account only after some sort of X min. So the lower boundary has to be discarded, okay? And again, we can see that the avalanche analysis in the LFP, now we look at the local full potential, and sometimes you see such behavior, and sometimes you don't see such behavior. So that's the interesting thing about it, that when you look at local full potential, you don't see that. And then when you compare the positive peak versus the negative peak, you see similar behavior, which is interesting because there's some sort of evidence that when you look at the positive peak, it should be different because the negative peak is associated with spiking. So if the system is organized by some universal dynamics that forces the system to behave in self-organized critical way, then therefore the negative local field potential, the negative peak of the local field potential should follow a similar characteristic as that of the spiking. However, what we see is that the spiking doesn't follow the power law behavior, but the positive peak and negative peak, they behave similarly, and sometimes they do and sometimes they don't. So this is a little bit more detail about such things, which I'm not going to go through. But in any case, so these are the main points of this study here, that the spiking and LFP behave differently. And LFP, even the negative local field potential, does not relate to what the spiking does. And what we also noticed that sometimes a multiple exponential fit can describe the system better, meaning that it is possible that you have two sort of a stochastic process in the system that they're behaving, and then they shut down suddenly. So there are some studies that they show that if you have such kind of system, then it could at times show a power law behavior. I'm going to talk about another part of the study, and that is the interaction between the two inhibitory and excitatory systems that I introduced a little bit earlier. So here you have two sets of recording. This is in monkey, this is in human. On the top you have local field potential, on the bottom you have a spiking activity. The ones in red are inhibitory, the ones in blue are excitatory. And they are sorted based on the firing characteristics within this epoch that you see. And you can look at different states of the brain. For example, you have wakefulness, slow wave, and REM, which is a dream. And down here, you have an aggregate of activity. So you basically sum up all the activities together, and you get these types of the red is the overall inhibitory activity, and the blue is the overall excitatory activity. When you normalize it by the number of neurons within a given system, then you can see they somehow follow each other. And this is for 60 seconds. This is about 10 seconds of such behavior. And the idea is that, so do we have two systems that basically try to control each other? Like when you have an activity, inhibitory activity, then the excitatory comes back, or vice versa. And they try to cancel each other. And if that behavior extends to different scales, temporal scales, okay? So for example, here, you can see, if you take into account about, so this is a given binning. So we bin the system at 32 different length scales. right? So those are shown at the very top. And it's a logarithmic increase of the scaling just for the computational efficiency. We had to just choose a certain number. And when you do have some sort of like coarse graining of the behavior, so this is 600 and something bin at a given, at the high, at number C1, you can see up there. So this is about 6 seconds, right? So each bin will be 6 seconds. And the aggregate of the activity of inhibitory neurons are shown in red, and the excitatory neurons in blue. And you can see they kind of like follow each other. And then if you take one part of it and zoom into it, now you change the coarse graining, you make it finer. You go to C2, for example, which is now 1.3 seconds. Then you can see, again, they follow each other, and so on and so forth. So you can go all the way up. And you can see that this sort of balanced activity is preserved across these different scales. So it's very interesting because Ken Wilson, actually, who introduced the idea of renormalization in systems with many length scales, showed that there is such behavior when the system has some sort of scale invariance across different states. And that one is in the spatial domain, but here we're doing it in the temporal domain. Okay, so that's the difference here. And when you look at the distribution of the sizes of activity, so now each column is one of those binnings, and as you go to the right, then you get to coarser and coarser time grains. And to the left, you have the fine temporal resolution. So on the up, you can see the activity, the distribution activity of excitatory neurons, and the bottom, you have the distribution of the inhibitory neurons, and you can see whether you're in wakefulness or slow wave sleep, there is some sort of preserved balance wherever you are. But that is an overall activity. When you look at the moment-to-moment transition, of course, that's not the case. So at any exact moment, you either have an excess of excitation toward right or an excess of inhibition toward left. But what's interesting is that the shape of this distribution, so these colors relate to different temporal scaling that I was telling you. So the blue colors are the fine temporal resolution. And then as you go up to the red, then you go to the coarser and coarser temporal resolution. So the distribution narrows down, but it kind of follows some sort of like symmetry that you can see in the system. And then one may argue, okay, but maybe this is all just like random incidents of stuff. So what we do is that we try to do some sort of cross correlation. Actually, maybe if I show you one animation here. Okay, so what you see here, so you start with one spike, and then you have some sort of, from one reference, for example, the excitatory one, and you have this kind of like binning around it, and then you have this box counting, and so for example, there's one here, so one gets added to this one, and it moves around. So this actually could move around, and eventually you go through all of the spikes in the system, and then you find out what's the behavior. If I can get out of it. Okay. So when you do that, you get all these kinds of, so there's a different states, different subjects, all kinds of things and different binnings. And always what you get some sort of very nice symmetry of the two systems. When you do some sort of randomization, then all this behavior vanishes. So this is one sort of randomization. For example, what you can do is that you can simply move the aggregates of the ensemble activity against each other. You can shuffle things locally. You can do permutation of the number of ways of doing randomization. In all cases, when you do randomization, this behavior vanishes. So in a sense, what we see here is a true representation of the activity of the network dynamics. And there's more that you can do. For example, what's interesting is that when you do a log, a probability curve, this is a probability curve, that you can see, for example, at the very end, you have deviations from the symmetry. So in a sense, system is showing this overall balance. But sometimes it deviates from the symmetry. And that actually happens to be the case more or less in the slow wave sleep. So when you have a slow wave, you have more deviation from the symmetry rather than the other cases. And it's possible because in a slow wave, you have some sort of like a toggle switch. You have up states and down states in the system in which you have moments of a high activity and then silence, moments of high activity and then silence. And these are due to some sort of like recurrent activation of excitation or inhibition with respect to each other. And then when we continue this, you can see, for example, there are cases that such balance breaks down. So this is an instance of seizure. So on the top, this is about 9 minutes. On the top, you have activity of excitatory neurons. In the bottom, you have activity of inhibitory neurons. And these are sorted based on the firing frequencies. And you can see somewhere here, some strange thing is happening, right here in the middle. Okay? And this is a zoom in into that part. It's about 40 seconds here. And this is the aggregate of excitatory activity versus the inhibitory activity. Okay? And you can see there's a moment that there is some dominance of inhibitory, which is very, very interesting because perhaps the system is trying to control whatever catastrophic event is happening. So, and when you look at it, this is just the raw binary matrix that you can visualize here. But when you also normalize it, and you can see there's a different scale, and this is the time within this period, this 40-second period, and you can see the same thing. This is just a zooming into it. So this is the top one, and this is the 40-second one. And you can see there is some sort of like multiscale breakdown of balance. So the balance breaks down when the system gets to seizure. And I'm going to go to the very top, talk a few minutes about different sort of okay. So now in the next few minutes, how much more I have? Like 10 minutes? Okay. So next few minutes, I'm going to talk briefly about what you can also see from behavior of system in a much larger viewpoint, for example. So when you measure the electromagnetism of the brain, this is like the first magnetic encephalography system that was actually invented at MIT, then you're looking at some around here. This is the range of the activity of the brain. So it's 10 to the minus 12 or 10 to the minus 13 Tesla. And then you have all these other kinds of noise, and you have to compensate for these noise within the environment by doing the experiments within a magnetic shielded room. But yet again, you also have the squeak noise. The squeak is the sensors, some sort of quantum user interface device that helps you measure the activity of the system. So when you do that, you have activities of the brain at different locations. You can measure EEG and MEG at the same time. The EEG is electroencephalogram, and MEG is magnetoencephalogram. And actually, this might be informative for you. So if this is how the neurons are shaped within the cortex, they're organized in the cortex, you have this intercellular current going down, and then you have a return current going out. So it's a diffusion of ionic stuff within the medium. So one side, which is this one, creates the magnetic field. And so you have to follow the right-hand rule, which means that the magnetic field sometimes is not visible with the MEG. If the orientation of the dipoles are like this, sometimes it's visible. But EEG is always visible, so regardless of where you are within the cortex. Okay, so this is just like some background about the signal. And when you look at the spectrum of the activity at a given channel, then you get this sort of, so this is the log frequency, and this is the logarithm of the power, and these are the frequencies as you go up. And you can see if you look at the low frequencies, then you get some sort of behavior that's sometimes it's power law, sometimes it's close to the power law, sometimes it's not close to the power law. But what happens is that when you take into account the signal-to-noise ratio for different sensors, then you can actually go through different ways of compensating the effects of the noise, because the noise could be coming from like the ambient noise in the environment. It's a white noise. It could be the noise due to the thermal characteristics of the squid sensor, which by itself actually gives rise to some sort of pink noise, which is similar to what we're seeing here. And when you do that, you repeat the experiments for different types, you can see that the magnetoencephalography, which is a system that is not influenced by the scalp, the muscles, the frequency filtering of the medium, all those kind of things, shows a very different behavior from the electroencephalography. And a lot of people have used electroencephalography to argue for the self-organized criticality and the kind of the organizational principles that I was talking to you before. More than that, you can also look at the distribution of the scaling exponent, of the slope of the logarithmic frequency power. And then you can see, for example, you get a very interesting shape for the MEG. And that's basically because the sensors around here, around the temporal lobe and the back of the cortex, they are more sensitive to the orientation of dipoles because of the folding of the brain and whatnot. And so this all tells us that somehow emphasizing too much on EEG, for example, when we don't know so many things about it, is detrimental, because we don't know the exact nature of it. It's being influenced, it's being filtered by the medium, and trying to extract some signature out of it and trying to say that the system follows the self-organized criticality is not very proper. I'm not going to go through the rest of this, and I guess we can basically jump to the end and try to summarize. So in a sense, we have a system which is composed of two excitatory inhibitory opposing forces, and they organize themselves in a balanced fashion. And they try to keep system in some sort of overall not very rigid point, but it is something that at fine temporal resolution, it can lead to computation. While if you take into account the larger temporal moments, then the system has to stay stable, basically. So if the system has to stay stable, it has to go also through plasticity. So the system is always changing. So you have a system which is always changing. It keeps itself stable. But perhaps for having this, the plasticity, it has to sacrifice something. And that sacrifice perhaps happens at the very molecular level, that it's not visible through here. That perhaps there's some energy expenditure that is not optimized over there or something like that. And with all that, I want to bring the presentation to the end. So these are the people who have been contributing to this research. For the theory, Alan Dehghani, who is the director of the European Theoretical Neuroscience Institute, and Claude Bedard. And then all the others were helping in the experiments. Some were giving us the data from UCSD, Harvard, University of Utah, and Chicago. And thanks for your attention.
Time for a few questions.
Okay.
So fast. I have a question. Okay. Of all the things you show, which one do you think is the one that is going to make the...
Well, I think the seizure is the most striking one, because it somehow shows that there are moments that certain principles actually break down, right? So there's an exact moment that you can recognize that something went bizarre. Because the problem with the brain is that it's really hard to connect what's happening in the underlying, architecture to what's happening on the surface. So a lot of people try to jump into cognitive neuroscience or system neuroscience, within which they record from different neurons, and they try to say, all right, so in such and so behavior, for example, you have an attention to the right, therefore you have an increased activity of your neurons. I think that's very informative from many points of view. But in giving us some underlying principle of the computation, the principles of the computation of the brain, I think that part is more informative.
If I understood correctly, you say you've observed a balance between excitatory and inhibitory activity.
Yeah.
Maybe I'm missing something, but it seems like such a balance is almost mandatory. Can you imagine a richly connected network of active nodes where either excitation or inhibition dominates, and you don't immediately go to one of the extremes of all the cells firing all the time, or a dead brain where nothing ever happens? I mean, is there a way to construct such a network where it isn't balanced?
Actually, it's a good question. Let me find something for you. Oh, actually, my mathematical has run out of. It wasn't mathematical, but it has run out of its license. Anyhow, so to answer your question, I did some sort of simulation. This was all based on data, right? So I did some simulation lately, which was this summer, and so I constructed different networks, so including different types of connectivity within the system, right? So ranging from random networks to small world networks to Barabasi network, all these different kinds of things. And there are times that you see, if you try to mimic to use the same number of neurons as is observed in anatomical studies, right? So it's always a ratio of 4:1. So when you do that, certain types of networks, they either, when you hit them, you randomly hit them with a certain number of activation in different locations, they move on and they die out at some point, okay? And some of them, when you hit them, they actually just don't move it at all. They're just dead from the get-go. They just activate for a few times, for a few cells in this. It was a cellular automaton. And then there are times that if you try to use a mix of kind of connectivity based on the the spatial-temporal dynamics that I told you. For example, we noticed that the excitatory neurons, they drop down, their connectivity, they drops down exponentially, but the inhibitory neurons don't. When you do that, that's the time that when you hit the system, it moves on much longer, actually. It stays active for much longer, as if the system is now capable of mimicking certain behaviors that are seen in the brain. But there was also moments that you can see such activity gets into similar to what I showed you in the last figure about the seizure, that there are moments that it actually stops, and you have a hiccup in the system, then it restarts again. If I can get it here, I can show it to you later.
Thank you.
You're welcome.
Nima, what motivates you to study this topic?
What motivates? Okay, so as I said, one of the things about a lot of people is that they jump at the cognitive part of the brain, right? That they try to say, all right, how do you connect the spiking of a given neuron to such and so activity? I do believe that's informative, as I said, but somehow it doesn't really give us the true view of how the system operates in the large sense, because we have to know, how do we have all these different states of system, right? You have sleep, you have wakefulness. And within all these, yes, the cognition happens within one part of it, right? So if we don't know what's the universal principles over there, then it's really hard to get into the core of the cognition. So that could be one answer.
All right. Thanks, Nima.
Thank you.